Hi, and welcome back to the EDGE tutorial series. This video is going to go over the PCR Primer Analysis module in EDGE. After this tutorial, users will be able to validate existing primers or design new primers based on a sample's assembly. As a reminder, we are running two job submissions in parallel. The first uses E. coli FASTQ files found in the PublicData testData directory in the EDGE interface. The second uses data from the SRA run SRR 2164314. EDGE supports both design and validation of PCR primers based on assembly results. Both of these modules must be turned on at the user interface. In the validation pipeline, users upload a file containing current primer sequences. The input file must contain an even number of forward and reverse primers listed one after the other, saved in FASTA format. EDGE maps these primers to the sample's assembly using BWA to determine if the amplicon is generated. Users must define the number of mismatches allowed in this validation. The default setting is 1. To design primers for newly assembled contigs, EDGE identifies unique regions of the sample using BWA, then generates primer sets using Primer 3. Users indicate desired primer reaction parameters for melting temperature, amplicon sizing, and number of primer pairs. We are going to validate a set of previously designed E. coli primers and generate new sets for the FASTQ sample. Since we don't know what we're looking for in the microbiome sample yet, there's no use for the primer tool to be turned on for this SRA sample. This is the last EDGE module, which means we're also ready to submit jobs. We will go over how to submit a completed job, check for errors, monitor job status, and look at project output. Let's hop over to the EDGE interface and finish up part 2 of this tutorial. Go ahead and turn the PCR primer analysis on for our E. coli sample. Before you do that though, let's go ahead and add an _2 to the name. This is going to be important because we already submitted projects at the beginning of this tutorial series with the same name. Scroll to the bottom and turn on PCR primer analysis. Under primer validation, part A, we need to click Yes to run the primer validation. Our primer FASTA sequences are also located in the PublicData testData directory. Go ahead and click on the circle button. Again, PublicData, testData. Scroll down and select the primer.fa file here. Maximum mismatches are going to be left the same. Now click on B, Primer Design. We also have to specify yes to run this pipeline. Let's go ahead and keep all primer parameters set as default. Job submission is as easy as clicking the Submit button at the bottom of the page here. Any issues or forgotten fields will be highlighted in red with clickable links and instructions on how to fix them. Here you can see that everything is saying it was submitted successfully in green. If you scroll up to the top left and click on Projects, there's a list of all the projects being run by a user. Projects are highlighted in orange if they are running, like you see here. They're red if they failed, green if they're complete, or gray if they're in the queue and not yet run. EDGE will run 3 simultaneous job submissions. Additional jobs will be started when computational space becomes available. The web browser does not need to remain open for this to happen. You can see the original samples we submitted at the beginning of the tutorial series. These samples were submitted with default settings only. We can compare the results in part 3. Like I mentioned, I added the number 2 at the end of the project with updated settings. We can go ahead and click on My Project list here. By doing so, users can see, search, delete, and rerun all the different projects. The top right button here also gives users a more detailed look at the current project running, including live coding logs for advanced users. You can see which step of the process is being run. For our SRA sample, we're also going to scroll to the bottom and click Submit. Make sure everything is green and submitted successfully. We can scroll back up and check that it's in our My Project list. Here, you can see that both of our FASTQ samples and SRA samples are running. This concludes part 2 of the EDGE tutorial series. Check out part 3 for an analysis of the output generated by each EDGE module.